Okay, so just a very quick kind of précis of what melanoma is and why we've had problems and why we've had to get involved in the discussion on value and why should any of us be interested in what other people are valuing our disease as. So basically, melanoma, my husband came in at stage 4 in 2011. Okay, before that, 6 to 9 months for metastatic melanoma was the norm. You died. Yeah, we had no drugs. We had some very old chemotherapies that didn't work, and these were our comparators. So this is why we had problems later on. We then had a nice line of targeted therapies. Half of the patients have a BRAF mutation, generally the younger half. So targeted therapies will work for those patients, but they won't work for people who don't have the BRAF mutation. So we then started on the immunotherapies. So we've had these for the longest. We've benefited and suffered from them. So we are actually quite a good disease area to understand the complexities of immunotherapies. So we had 4 reasons why we started getting involved in measurements of value. And the first one was that our drugs were proved so fast that the trials were very small. It was unethical to leave people on a standard therapy that was bad for us. So we moved very fast. We had trials that were too small, 800 people, and we know that HTA assessors and payers, they like 8 to 10,000 cases. So we were talking about 800 people on these trials, which finished early. Also risk. You know, if you're a trial that only has 800 people, there are many side effects that don't show up. The 1 in 1,000 side effects, which are actually quite common, don't show up in these trials. So there's no record of this. And we were kind of suffering from this really fast progression of new therapies coming in, suffering and benefiting, obviously. And I just like this quote from Nicolas Moore, who's a French specialist. Absence of a demonstration of risk is not a demonstration of absence. Yeah? Just because we don't have the data doesn't mean that it doesn't exist. Okay, the second reason that we wanted to get involved was that we kind of felt that we were being a little bit manipulated and maneuvered into a certain view on value frameworks. We had no clue about it. We didn't even do our homework. We didn't really get involved before 2015, let's say. And we found industry was kind of telling us how value frameworks, these are not good tools. They really don't involve the patient view. The perspective hasn't been sought. These are not good tools. So we thought, well, we'd actually like to make our own minds up about this. Similarly, HTA, who've been doing value frameworks forever, they didn't know why we would want to be involved. You know, leave it to us, we'll look after you, we make the right decisions. And we were finding that our decisions, the decisions being made by HTA and then the payers, were not suiting us. We weren't getting access to drugs because of various reasons of our small trials, and we wanted to make the decisions ourselves. So we were asked by BMS, the BMS goal meeting at ASCO last year, just to do a little précis of the magnitude of clinical benefit scale that ESMO created in 2015. And we'd only just started dealing with them, and we'd basically gone to them with kind of a slightly aggressive attitude saying, why aren't patients involved? And although our belief is that if patients aren't involved, it's a missed opportunity and it's a shame, but it doesn't necessarily mean that the tool is bad. So we wanted to assess this properly. And we approached ESMO and we said, okay, you know, why are you excluding patients? And they said, we're not. We'd love you to come in. We're doing version 0. We're going to do version 1 in a short time. We'd love people to come in and tell us what's wrong with this. These are real nerds. Yeah, they love their product, but they really want it to work. So we did a little paper just for this meeting, basically saying that our experience with the value frameworks was not quite what we were being told we should believe. Okay, so the last thing that we think this is so valuable for is patient education. We all need to learn about science. Yeah? We cannot avoid it. If we want to be able to argue our HTA point of view, if we want to argue with the payers as to why we're not getting drugs, we need to know our stuff. And it's hard because you have to educate yourself in areas that many of us have never even been involved in. But this is important. You know, it's important for individual patients and as advocates. Yeah? We need to, the evidence is what we want. We want evidence that our decision makers are making decisions on our behalf. Yeah? We don't want eminence-based science. We don't want to go before a doctor and have be told, this is the strategies you should follow. Yeah? That's eminence-based science. We want the evidence. And also, it protects you. We always say that, you know, knowledge is protection. It's power, but it's mostly protection. So we need to have ways that we can actually assess these tools from our perspective, as well as everyone else is doing for us. Also, I think we can't actually underestimate that you cannot influence what you don't understand. So if you're being told this is the value assessment that we've made of your drug, if you don't understand it, you can't argue it. You can't say where the flaws are or the weaknesses are. So this is vital that we use this very much as an education tool. So when we made this approach to ESMO, we actually had a great feedback from them, and we said, okay, we want to train our people. We want to train our people to read clinical trials, papers, and we want to train them how to use these scales. And they said, cool, no problem. So, first of all, there's a whole rash of value frameworks came out, and mostly from the US, but a few in Europe. But Europe had been using it in the HTA process for ages. So value frameworks, they just are measuring value, but value, it totally depends on your position. What I value is totally different from what any of you will value. What a payer will value, obviously, is totally different. What HTA will value is totally different. So all of us have different values. How on earth can you actually bring that all together into one concept of value? It's pretty much impossible. So the independently developed frameworks were basically a counteract to the kind of HTA, slightly secretive, this is how we make our decisions. So they were more academic, and they actually wanted to also allow oncologists in the US, for example, to sit down with a patient and actually value a certain treatment. And in some cases, they are independent, but they're actually sharing the same procedures, if you like QALYs, that HTA are using. And many of us in the patient community don't like QALYs. It doesn't reflect our values. Okay, so it directly affects us because decisions are made on our behalf because of them. Yeah? We need to understand them. So here we're bringing these frameworks, so the scientific concepts, incorporating variable components which are considered valuable for each group. Yeah? Components are not absolute, but with a certain degree of subjectivity. What's the value of innovation? Some countries will value innovation more because they actually make lots of money out of pharmaceutical industry within their country. Yeah? How do you value unmet need in a country? Small disease areas, lymphoma, the subtypes of lymphoma. How do you value that? How do you value the burden to a population of the cost of new drugs? Yeah? How do you value expert opinion? We will look at the ASCO framework. They use a lot of expert opinion in there. And for me, that's one of their weaknesses. It's great, but we still want it to come from evidence. So this is just some of the frameworks that have been developed. We had some very early, so the Netherlands had one in 2009, which was basically a costing tool, but they costed the drug and not the whole treatment. So that failed in terms of, you know, the impact on a country's budget. The NHS Cancer Drugs Fund prioritization tool, yeah, 2011. First of all, it only prioritized cancer. The selection of drug was not transparent at all. They could choose who they wanted. And lastly, it undermined the HTA decision. So NICE said, yeah, we reckon this is a valuable drug, and they could just cut it from underneath and say no. And this is the kind of situation that we've also had in France, where our drugs have been cut from underneath us, even though the value frameworks have said this is promising. Then Memorial Sloan Kettering, which is the big hospital in the US, did a kind of funky tool called the Drug Abacus, which I'll show you shortly. The ICR Value Assessment Project, these are all US ones, NCCR evidence blocks, ASCO Value Framework. Now, all of those top ones have cost in it, yeah, cost of drugs. Now, we know that the pricing of drugs in Europe is a holy cow. That is kind of, the decision is held by the countries. So the ESMO magnitude of clinical benefit scale doesn't bring cost into it at all, which is kind of why we like it, because we know it's going to go into the different countries and have different decision making based on budget impact and all kinds of things that they make decisions on. So this is the drugs abacus. And I would recommend that you just go and look at it, put your drugs in there, have a little play around with it. It has a whole lot of little modifiable components at the bottom here. So you can have, like, if your country has a threshold, a quality threshold, over which you have to have had some benefit in 1 year, you can modify that. So I think in Belgium it's like 45,000 euros a year. In the UK it's slightly less. Different countries have different thresholds, but you can play around with that. You can also then have a toxicity discount. So if you're not prepared to put up with toxicity, depending on what other standard treatments you've got in your disease area, you can change that. You can play with all of these things. And I've left up there ipilimumab, which is our anti-CTL4 immunotherapy, which is our first immunotherapy. And you can just see the difference. The top is the actual price, and the red dot below is what the scale thinks it should be worth. So in a way, you can see why industry wasn't 100% in favor of these things, because generally it undervalued from the prices were being asked. It generally undervalued them, partly because of lack of evidence, but for various reasons. So, you know, this is why you get pushed in one direction, and HTA were doing the same, but from the reverse. So this is quite a good tool. So go and have a little play with it. The ICR Value Assessment Project, also US, also has the drug prices in there. It has a arbitrary. The limits is that there has an arbitrary limit on the amount spent a year. So that doesn't really apply so much to us Europeans. And also the value is based on QALYs. So if you don't like QALYs, the quality of life years that NICE use and various other countries use, you know, this is not really the thing for you. So then we had the NCCR evidence blocks, where they basically give a score on the efficacy, the safety, the quality of evidence, consistency of evidence, and the affordability. So again, the price is in there. The problem was that the choice of the units was not very transparent, a little bit similar to the Cancer Drugs Fund. So nobody really knew how they were making the decisions. Okay, and then we had the ASCO value framework. Now, the nice thing about the ASCO one is that, like the ESMO magnitude of clinical benefit scale, you can actually do it yourself. You don't have to have complex workings out. You don't have to have any prior knowledge. You can just do it yourself. But again, they have cost in there, and the quality of life is excluded, which from a patient perspective is unfortunate, let's say. But it's also worth having a look at. But you know, since it's not up to us what the prices are, the cost, you know, they're discussing a doctor and a patient in the US trying to come up with their best strategy. So it doesn't so well apply to us. Okay, so this is the ESMO magnitude of clinical benefit scale. It's very simple. It's just efficacy and toxicity, yeah? And everything comes from the pivotal clinical trials, yeah? All the evidence. And you have to be a good enough clinical trial to be used on the scale. So, and it basically, all they wanted is that across Europe, decisions were being made differently. So one country was approving, even though the EMA had said, the European Medicines Agency had said, yes, we approved this drug based on its efficacy and its toxicity. Then they go out to the countries, and the different HTAs were making different decisions. So they were trying to give a tool that was independent of the HTAs, so that other stakeholders could actually make a decision based on the same kind of thing that EMA bases their evidence on. So you have curative settings, and you have non-curative settings. So if the drug is going to cure you, so here it gets approved by EMA, you go and look at it, is it a curative one? It's graded from A to C. Yeah, and also in adjuvant. So in the adjuvant setting, where it's disease-free survival, that's also a curative setting potentially. So they're kind of in between for the adjuvant therapies. For non-curative, so things that are just prolonging your life, they have a different set of papers that you fill in, and they're graded 1 to 5. And it's kind of similar to some of our HTA processes. In the curative setting, the measures they use are overall survival, progression-free survival, the two golden standards. Now, that's pretty difficult if you're a rare disease. It's pretty difficult. I mean, even with us, we weren't getting that evidence. We also had some problems in that progression-free survival in immunotherapies is undervalued. It always used to be considered as it's always overvalued, so we don't like it in the assessment process, but generally now we're seeing that progression-free survival in the immunotherapies is undervalued, so it should bump up our scores. In the non-curative, so you have OS, PFS, and then quality and toxicity outcomes, so you can add in various things. And now they've even got the third form, which is for outcome measures for like rare diseases, so response, overall response rate, things like that, which generally they don't like, but they've actually brought in a form to actually allow these things for the rarer disease setting. Okay, so the elements that they use. So obviously cost down the bottom, that's not in there at all, which it is for the other scales. Prognosis of the condition, long-term survival, overall survival, quality of life, toxicity, all of those come into the scale. And when we talk about how we use these scales and how we actually applied it to our disease area, we'll go into that a little bit more. The main problem that we found was that because our trials had been so small, there were subgroups who actually reacted really well to a treatment that didn't even come into the scale because it does it across the board. So we had treatments refused that really did work for some subgroups that were just cut out because they didn't score highly enough, and they will never score highly enough because we can't go back and recreate evidence if in the clinical trials they haven't done subgroup analyses or that it's been underpowered in some way. You're never going to get that evidence until you've got enough real world data coming in, and that means registries and more trials. So, but that stops patients getting drugs, and we don't want that. So this is some of the principles of the MCBS. So cure takes precedence over the deferral of death. Yeah, that's more important. Direct endpoints of survival, quality of life, take precedence over surrogate ones, PFS and RR. So, you know, this is where we should be in there, because in rare diseases, you're not going to have the trial, you're not going to have the powering to be able to get overall survival for ages. So you need to be knowing what they are looking for in order to be able to argue your point. Disease-free survival and curative disease more valid than PFS or response rate in non-curative disease. Interpretation of the evidence for benefit derived from surrogate outcomes, such as PFS, may be influenced by secondary outcome data. So they do add in caveats and things. So let's look. Okay, so these are the scales. So this is the curative setting. So if your drug is something that will cure you, it's valued A, B, C. C, it shouldn't really be in the formulary. And us as advocates, if we want to get these good drugs coming in, new drugs, innovations, we should also be arguing that the bad drugs should no longer be on our formularies. That's part of advocacy, it's two-way. On the non-curative setting, you have 5 and 4, those are the good ones. Our drugs were valued at 2 initially. We knew they worked, we knew from the phase 2 trials that they worked, and yet our drugs were valued at 2. And that was purely because of a lack of evidence. They didn't have the numbers, and they didn't have the subgroup analyses, and this is a big problem for us, and this is one of the main reasons that we got involved, because you need to argue your point. Then we have, can you compare these scales? Now, it's pretty difficult to compare them because everyone has their own interest point when they develop their scale. We can see that we don't have costings in there for the MCBS, but they're all pretty dynamic. And I just wanted to put this up because we have an input in here. Yeah? We can actually change the development of these scales by knowing enough about them and arguing our point as to why certain evaluations are wrong. Yeah? We mustn't be scared to come back to them and say, this isn't counting on your scale, you're undervaluing this drug because of a subgroup that it works very well in, that is undervalued because it doesn't work well in other people. And we don't also have any idea of who it's going to work for. So this undervalues our drugs. So we need to be able to argue these points. So this one I just put up because it's for multiple myeloma. Now, it's done by the National Pharmaceutical Council. Yeah, so they've compared it. Be very careful with comparisons that you find online or in the literature, because it depends who's doing it. yeah, and whether they think it's something that shouldn't be there or whether, so the slant is always different, but it's really good to see. But it's also, you know, that's myeloma, so there's blood cancers here. So even though the scale has not really been used in blood cancers until very recently, it's only been in solid tumours, this is coming, so this is why you guys need to be ready for it. So this is where we really benefited from getting involved in this. We had our conference this year, we had 120 people, and we said to ESMO, we want to train our people in how to read clinical trial papers, because that's the fundamental starting point of then using these scales. And they were fantastic. They printed us, I don't know how many, we told them that there were 70 people, so we needed groups of 4 or 5, and then actually there were 120 people, so they freaked out a little bit. But they printed out clinical trial papers from all solid tumour areas, and we just put them in front of our patient groups, and we said, okay, here is the blank magnitude of clinical benefit form. You need to go through these clinical trial papers, find the relevant outcome. What is the outcome? What is the hazard ratio? Is it a toxicity? What are the toxicities? You go through that paper and find out the pertinent things that are going to be used in a scale. And ESMO was quite worried because they didn't think patients were capable of this, and most of our people were stage 4 patients. So, but they were kind, and they really bent over backwards to help us with this. And we had a fabulous day. And you know, the feedback we had from the patients was, this is what I need to be learning. This is real science for advocates. Now I understand why decisions are made in our healthcare, and how they decide, and how difficult it is to decide, and how we cannot make a decision based on 2 clinical trials that were underpowered, and that's how it's actually affecting me, because I know I don't get the drug. It was really, really important. And it's basic science and basic statistics, but really a worthwhile training exercise, and we're going to do it every year in different aspects. So this is the thing that we wanted people to realize is, why is it necessary to go to the original scientific literature? Yeah? And this is why, okay? This is what everyone else is making decisions based on. Yeah? We need to know this. Yeah? Scientific publications, they appear before anything else. So you're current when you go to the original source. Yeah? And I can tell you, I've had many discussions with oncologists who have not a clue what a value framework is. I had a discussion the other day with an oncologist who didn't know the difference between a regulator and a health technology assessor. Okay? So don't expect that your physicians know how to value drugs. They do not often. Some do, but they often do not. Correctness, yeah? So we're always reading rubbish data out of the local newspapers about how cancer's about to be cured, yeah? It's misleading, yeah? We need to go to the source of the data and not just accept it as it's been regurgitated third hand. Empowerment, yeah? We have to take responsibility because this is affecting us. Yeah, everyone else has their own value reasons. This affects us. This is live or die for us. Yeah, so it's extremely important that we know what we're doing here. And I think a good read is Ben Goldacre's Misreporting Science. It's worth a good start. So we started with how to read a scientific paper. So I think I stole this from Bettina. I don't know if people know Bettina Ryle. She's a kind of a force to be reckoned with. She's also the chair of the ESMO Patients Advocacy group. And she has this, my personal recipe of how to read a clinical trial paper. And I think it's absolutely spot on, because everyone goes to the summary or the conclusion, because it's quick. You don't have to read the whole thing. And that's kind of the worst thing, because that's really what they want you to understand from their data, without actually saying this proves, that they are misrepresenting themselves. So read the abstract, what it's about, go to the figures, what does the data look like? Who have they excluded? Yeah, what subgroups? Have they only chosen the most healthy patients for their trial? Yeah, that gives really good results. Read the results section. Yeah, and have I actually seen that in the figures that are reproduced? Read the introduction, if you've managed to resist until now, yeah, because that's what everyone does. go back to the results in case I've missed anything. And if you know the scientific method, it's always nitpicking. You're not trying to find what's right about the paper, you're constantly trying to find what's wrong. And if you cannot find anything wrong anymore, then it's pretty much probably okay. Yeah? But the people who write the paper will be telling you, this is the best piece of research we've ever done. You should just take it at face value. And at the end, this is the conclusion we always have. You know, we're going to save the world. Cancer is cured. We can't be accepting that. Making up your own mind is also really important, but this is part of your training. Do it several times. Do it on different papers. Do it on different disease papers. See if you can do the same thing. It's very good training, and it does take time. And this is just because I really believe that the only people who change anything are groups like us. Yeah? Everyone else has their stakehold. Yeah? But it's the only thing that changes things is people who are committed to it, like us. And that's it. I hope that helps.